Good afternoon, everyone, and welcome. I'm Jack Van Horn from the University of Virginia. It is my distinct pleasure to welcome you to this edition of our 2024-2025 Foundations of Biomedical Data Science Seminar Series. Our unique lecture series is made possible through the generous support of the Integrated Translational Health Institute of Virginia, the UVA College of Arts and Sciences, the School of Data Science, and through a grant from the National Institutes of General Medical Sciences. Since its founding in 2016, the Foundations of Biomedical Data Science Seminar Series has been at the forefront of exploring the transformative impact of data science methodologies, artificial intelligence, deep learning, and statistical modeling across diverse themes in biomedical and health sciences. Our selected theme for this year is focused on quantitative approaches in spatial multiomics for guiding personalized medicine, where every Friday we enjoy presentations from a wide range of leading thinkers about the issues, the promise, the opportunities, and the hurdles associated with advanced transcriptomic, genomic, metabolomic, and other omic methods across a wide variety of data analytic opportunities which they present for data science, deep learning, AI, and spatial modeling. We are very enthusiastic about this year's Biomedical Data Science Innovation Lab program, where junior faculty, biomedical, and computational experts will develop joint and novel projects on the topic of quantitative approaches and applications for spatial multiomics data types and their use towards guiding personalized medicine. Participants selected for our special program utilize the content from seminars such as these as essential material for our culminating in-person manuscript and grant project development workshop to be held this year at the Hutton Hotel in downtown Nashville in June. We are very excited about today's lecture. It represents the 150th Foundations of Biomedical Data Science Seminar Series lecture. This is a major milestone for us. We've been absolutely delighted to have brought you these lectures from experts in the biomedical and data sciences, and we thank you for joining us each week and over the years, and we look forward to continuing to do so for years to come. I want to give a special shout out to Gina Kamdar and to Hannah Kears, who worked behind the scenes to pull all this together and to bring you this great lecture series. It would not be possible without them. For today's 150th seminar lecture, I am delighted to welcome our speaker, Dr. Chongzhi Zang, who is here at the University of Virginia in the Comprehensive Cancer Center here in Charlottesville, Virginia. Dr. Zang is an associate professor of genome sciences here at the University of Virginia and director of computational genomics at the Comprehensive Cancer Center. He is a computational biologist with expertise in cancer epigenomics, and his research focuses on algorithm development for high-throughput genomic data analytics, integrative modeling of gene regulatory networks and mammalian cell systems, and he is a past participant in the BDSIL program, a past mentor, and he will be a mentor again for this year's Biomedical Data Science Innovation Lab. In the spirit of our 2024-2025 Biomedical Data Science Seminar Series theme, Chongzhi's lecture today is entitled Modeling Single Cell and Spatial Omics Data to Study Gene Regulation. Gene regulation plays a central role in a wide variety of biological processes and in human diseases, and advances in single cell and spatial omics related technologies allow for high throughput, high resolution measurement of gene expression profiles, yet it remains challenging to unravel how cell type specific transcription programs are regulated. His presentation will focus on his lab's recent work in developing computational methods to predict active transcriptional regulators using single cell and spatial omics data. His work will demonstrate the power of integrating computational modeling with cutting-edge omics technology to deepen our understanding of gene regulation. As always, we are streaming this lecture live and for recording via YouTube. If you're watching on YouTube, thank you so much for joining us. Also, our specially selected 2024-2025 Biomedical Data Science Innovation Lab participants, as well as alumni, are encouraged to submit any questions they have via the chat feature in their YouTube sessions. I'll be synthesizing these questions and asking them on your behalf in the last 10 minutes or so of the lecture. And with that, Chongzhi, thank you so much for giving our 150th seminar lecture. We are really looking forward to your presentation.
Thank you so much, Jack, for the very kind introduction. And hello, everyone. It's really my great pleasure to take this opportunity to give the 150th lecture and to share with you my lab's recent research on developing computational methods to study gene regulation using single-cell and spatial omics data. So over the past few decades, the emerging genomics technologies has really enabled us to study genome functions in a more quantitative way. Using these genomic assays, people can transfer biology into data. As a computational biologist, my job is to reconstruct the data, reconstruct the biological knowledge from the data. So my lab uses two types of approaches to study gene regulation and chromatin epigenetics. So on the one hand, we develop computational methods. So computational methods include different levels. A fundamental level of computational methods are the quantitative models. The second level is the computational algorithms, and on top of this is the third level, which are we also implement these algorithms into software tools so that the community can use the method we developed in their own research. So here are several computational tools that we develop with different goals of studying gene regulation and analyzing high-throughput genomics data. On the other hand, we also use the computational methods we develop and other data science approaches to make biological discoveries by integration of big data that are publicly available. So we're very excited to make new biology findings simply by innovative modeling of existing data as a new paradigm of biological and biomedical research. So over the past decades, we witnessed a significant advancement of single-cell omics technologies, which revolutionize our understanding of biological systems. Single-cell data are multidimensional, as they provide multiple modalities of information of a biological system that usually composed of a heterogeneous cell population. Depending on specific technique, this information being measured in single-cell technology could include cell surface protein markers and intracell cellular information such as RNA transcriptome using single-cell RNA-seq, or even intranucleus information such as the chromatin accessibility profiles using single-cell attack-seq, or a combination of multiple modalities such as 10x Genomics single-cell multiome, which can measure RNA and attack from the same cell in one assay, or Cy-seq, which measures surface protein and RNA profile from the same cell in one assay. So over the past few years, my lab have been collaborating with experimental biologists to use various types of single-cell technologies to study chromatin and gene regulation. Just to give you a few examples. So in collaboration with my colleague here at UVA, Dr. Clint Miller, who is an expertise in cardiovascular genomics, and we use single-cell attack-seq technology from human coronary artery samples from over 40 patients with different stages of coronary artery diseases, and my postdoc Sean Hu analyzed the data and generated a single-cell or single-nucleus atlas of various cell types from human coronary artery tissues. And we can identify different subtypes with different chromatin accessibility profiles from these samples. In another collaboration with Dr. Taki Kim from the University of Toronto, my graduate student Hongpan Zhang and my postdoc Sean Hu analyzed the single-cell attack-seq data from a mouse embryonic gut tube. So this project, we wanted to show that using single-cell attack-seq alone can reveal different cell types or tissue types. So in early mouse development at E9.5, when the embryonic gut tube is not differentiated into different organs, and we cannot observe morphologically from the gut tube that which cells will go to develop into which organ. But the cells in the gut tube already show some chromatin accessibility features measured by attack-seq that indicates which organ this cell will develop into. And very interestingly, we observed on this dimensionality reduced visualization using UMAP that we can see the single-cell patterns that we predicted to develop into different organs clearly reflects the organ fates as well as their relative anatomical relationships along the body, from esophagus, stomach, all the way down to intestine. In another collaboration with Dr. Todd Bauer here at UVA, we are interested in using single-cell multiomics to study the heterogeneous pattern of cancer. So we use we are interested in study pediatric cancer metastasis, and we use a mouse xenograft PDX model and collected samples from both the primary tumor as well as the liver metastasis. And using this single-cell multi-omic data, we can observe the different cell type patterns from both the PDX tumor cells as well as the tumor microenvironment and mouse cells that are composed of different cell types, including many immune cells in the tumor microenvironment. So all of these single-cell technologies generated all of the data reflecting different cell types from single-cell from different biological systems. A common question we're interested in to address is that how to study gene regulation from such single-cell omics data, and specifically what transcription factors can regulate a gene set of interest or a specific genomic features. So generally, single-cell data, but when analyzing single-cell data, we generate different clusters of cells which we believe to be different cell types or different cell states. And these after annotate these different clusters, we can generate cluster signature genes or marker genes from each different clusters. Just like when we do differential gene expression analysis by analyzing bulk RNA-seq data, these identify the differential expressed gene sets or signature gene sets. They are co-expressed in a certain biological condition or biological cell type. And we are interested in whether there are some common or shared transcription factors that regulate such gene sets. So because we do, other than a list of genes that are co-expressed, we do not have other information directly from the single-cell data if it's a single-cell RNA-seq data alone. So we wanted to ask, how we're going to identify ways to see how we can predict transcription factors. So how to predict or identify functional transcription factors? There are different types of existing approaches, such as using ontology-based approaches, which uses the existing knowledge from different databases. And of course, they are limited by the knowledge or information from existing databases. We can also use co-expression-based approaches, whose fundamental assumption is that to look for the co-expression pattern between the regulators as well as their target genes to identify the potential transcription factors that are active. However, the expression of a transcription factor itself does not always mean the regulatory activity of that transcription factor. And I will show you some example later in this lecture. Of course, because most transcription factors have a DNA binding domain that can specifically recognize a DNA sequence motif in the DNA, we can also use DNA sequence motif based approaches to look for DNA sequence features that usually representing the sequence motifs or motif occurrence of transcription factor binding sites. However, the same motif may be interact by different transcription factors, and the simple motif occurrence does not necessarily mean transcription factor binding. And also, it's difficult to tackle for distal enhancers because it's difficult to identify where are the active regulatory elements in the genome that regulates the gene sets of interest. So over the past few years, my lab has developed a series of computational tools. We named it BART, standing for binding analysis for regulation of transcription. So BART can identify transcription factors from various types of input data. And we wanted to take an alternative approach that complements the existing approaches. Specifically, we take advantage of the large amount of publicly available ChIP-seq data that directly measure the genome-wide binding profile of different transcription factors, as well as histone modifications that quantify the potential enhancer or regulatory regions activity of different regulatory regions in the genome. So we do not use the transcription factors expression information itself, and we don't use the transcription factors sequence motif either. But by only using these public data collected from the public domain and mainly in the Cistron databases, and we developed this model. The idea is to use that we collected a cis-regulatory element repertoire in both human and mouse genome by using the chromatin accessibility data collected from hundreds of data sets. So in human genome, we have over 2.7 million potential regulatory regions in the genome. And then we use H3K27 acetylation, which is an active enhancer histone mark, to quantify the enhancer activity level or regulatory activity level on each regulatory element, as well as around potential interesting genes. And also over 13,000 ChIP-seq data that measures the genome-wide binding profile from hundreds of different transcription factors in different biological conditions. So using the integrative modeling, the input data can be simply a gene list that we're interested without any other information. And then using by integration of public available data, we are able to predict which transcription factors that have the genome-wide binding profile that most similar to the predicted quantitative regulatory profile that regulates the target gene set. For other type of input data, such as a tag-seq or ChIP-seq or even just a genomic region set, we can directly map this information to the cis-regulatory element repertoire and associate it with genome-wide transcription factor binding profile. And we also developed another version to analyze 3D genome profile data or high-C data to identify potential transcription factors that are associated with differential chromatin interaction regions. So this work was spearheaded by my previous postdoc, Zhenjia Wang, and previous visiting students, Wenjing Ma, and we have published this as a series of tools as well as a web server so that people can use BART-2 to analyze their data. And we're happy to see that other researchers have used BART to find important transcription factors in various systems, such as in cancer systems, as well as in immune systems, and they make their interesting discovery and published their findings in a few Nature papers. Also, we're very excited to see that BART, the tool that we developed, ranks on top in terms of different metrics of performance from an independent benchmark study recently. So this BART-2 is really a powerful and good performing tool for predicting transcription factors from the data of interest. So now, how to predict these active transcription factors from single cell data? So that's our main topic for today. So we actually see that others already directly applied the BART method to single-cell RNA-seq data to identify transcription factors from their data sets. So this is a paper published a few years ago in which the authors used BART to identify specific transcription factors that are regulating different subtype of tumor-associated macrophages. Although BART was not specifically designed for analyzing single cell data. So now, my graduate student Hongpan Zhang took this project to develop an updated method of BART. We name it BART for single cell, or BARTsc. This method is specifically designed for analyzing single cell data, and with a similar goal of identify active or functional transcription factors from single cell data, and with the various and flexible input of different types of input possible. So the input can be either just a single cell RNA-seq data or only a single cell ATAC-seq data, that they can also take in input of the single cell multiome data that have single cell RNA and single cell ATAC from the same system. So or we can use this commonly used existing tools to identify cell clusters from this single cell data, and from these cell clusters to extract the features, or namely either the cell type signature gene list or cell type signature chromatin accessibility regions. And then we also try to identify pairwise. We can also do a pairwise analysis comparing each pair of identified cell types or cell clusters, and identify the pairwise differential features that show either the differential expression or of genes, or the differential accessible regions from ATAC-seq, that comparing each pair of regions. And then to use this information as the input of original BART. So if it's only RNA input, so that the input can be just a gene set using the similar idea from BART, or the ATAC-seq regions as input to directly associate them with the potential regulatory repertoire, or if it's a bimodal input, we integrate these two information from there, and similar to the original BART, to associate with public transcription factor ChIP-seq data sets, and to generate potential transcription factors that are either active in each specific given cell cluster or cell types as a feature of cell type signature analysis, or a cross-cell type analysis, in which we can do the pairwise analysis to identify each transcription factor's relative activity, comparing each pair of cell clusters or cell types. So we test our BART single cell method first using a system in mouse brain development. It's a single cell RNA-seq data, and we show that for the each identified annotated cell type, we can identify the specific transcription factors that are active or functional in each given cell type, and our identified signature score is much higher on these functional transcription factors compare with other transcription factors for each of the cell types identified in this system. And next, we used another publicly available single cell data that are from single cell multiome co-assay of RNA and ATAC-seq to show the performance of BARTsc in analyzing single cell multiome data. So in order to quantify or evaluate the performance, we have to have a ground truth or underlying ground truth of which transcription factors are functional in which cell types. So in this PBMC data, we have 5 large different cell types, including B cells, T cells, natural killer cells, NK or monocytes, as well as dendritic cells, DC. So we collected using the literature to collected confident key regulators for each of these subcell types, and use this as the underlying ground truth or golden standard to see whether we can identify the correct transcription factor from each of the different cell types. So first, we show that by using the bimodal mode, or using take advantage of the multi-ohm pattern, we can show an improvement of identifying the key regulators from each for each cell type. So this is the this slide shows the percentile rank of the actual key regulators for each cell type. And if we use RNA mode only or ataxic mode only, compare with we use the bimodal mode, and we show that the bimodal mode can show an improvement in terms of the rank. The rank will increase for many transcription factors in the system. So in addition, we want to show that BARTsc can characterize the relative transcription factor activity across different cell types. So as I mentioned, we can generate the pairwise differential features or pairwise differential expressed genes by a pairwise comparison between each pair of cell clusters, and use BARTsc to generate this metric. We call this deviation ratio that quantify the relative activity of each transcription factor comparing each pair of cell clusters. And then we generate an average or mean deviation ratio that quantify the transcription factors activity in each given cell cluster comparing to all other cell clusters to give a relative activity measure. And using this approach, we show that we can indeed quantify or identify the active transcription factors across different cell types and even across different subcell types in the PBMC data. And then we compare our metric generated from BARTsc with the normalized relative expression level of the transcription factor itself, and also the occurrence or enrichment of motif of each transcription factor. The idea is to show that using our tool BARTsc, we can identify active transcription factors that may or may not be associated with the expression level or the motif enrichment of that transcription factor itself. So in this example, we show that SPI1, which is a myeloid lineage commitment key regulator that should be active in all myeloid cell types, even though in some cell types the expression level itself is relatively low. And just here are a few other examples that we can identify the active transcription factors in certain cell types whose motif activity may not show such specificity, such as the KYL4 in monocyte, which is a monocyte marker transcription factor, and BCL11B, which is a T cell marker, but they do not show the motif enrichment from this picture. And the IKZF1, which is a marker for both dendritic cells and B cells, and we can observe the activity from our tool, but they do not show the expression specificity. So, which means even they are overall expressed in all different cell types, the activity of this transcription factor is only they only work in B cell and dendritic cells. So then we try to integrate different dimensions of information to generate a integrated or overall prediction of active transcription factors or key regulators from a single cell data sets. So we use three metrics or information from three dimensions. So one is signature contribution, that is the measure the transcription factors contribute to the signature gene expression of one cell cluster or one cell type. So that's only looking at one cell type at a time. And the second dimension is relative activity, is to see that whether a transcription factor is relatively active in this given cell type comparing to other cell types from the cross cell type analysis. And the third dimension is what we call the uniqueness, is whether to see even if a transcription factor we observe is relatively high in this given cell type and comparing to other, and whether this factor is the only is the factor that only highly active in this cell type, but not other cell types, and also much higher than any other cell type. So to quantify their uniqueness. So after combining this three-dimensional information, we can generate a final list of predicted transcription factors ranked which are ranked high in the list in BARTsc score, that these factors we predicted to be the key regulators or key active transcription factors in each cell type or each cell cluster. So how to validate that whether we already have the ground truths from each cell types, and we just compare to these our prediction with the ground truths of transcription factors for each cell type, and we can quantify the performance of our tool compared with several other existing tools that use different approaches to analyze single cell data to generate transcription regulation. So several existing tools that we compared includes Scenic Plus, which is a popular tool for analyzing gene regulation and regulatory network from single cell data. And we use Scenic Plus have both the gene mode and the region attack input mode, and we compare both of them, as well as several other methods that can analyze transcription regulation from single cell data. And by comparing with the performance on the specificity and sensitivity or integrative F1 score of predicting the true transcription factors for each of the cell types in PBMC, we can see that BART single cell is the best performing method in most cell types comparing with other methods. And then we also want to evaluate the performance of BART single cell on a cancer single cell sample. So we used recently published the data of single nucleus sequencing of benign pancreas and plus pancreatic cancer data sets. So they are 10x genomics multiome single cell multiome data that have a tag-seq and RNA-seq together. And we use their paper, their annotation of each cell type from their publication, and we wanted to see what transcription factors we can identify from each of the clusters. Now this heat map, we can see that we can identify the key regulators and are specific to each of the sub cell type from these complicated pancreatic cancer single cell data. And similar to the previous case study, we use their published reports as well as other literature to find the true transcription factors that are active in each cell types in this system. and we want to evaluate whether BARTsc can successfully predict each of these transcription factors from each of this cell type. And we can see relatively speaking, our BARTsc tool can predict the most of these factors that are ranked on top among the percentile rank list compared to other similar methods. So this shows that indeed BARTsc can predict the known or the true key regulators from analyzing the heterogeneous single cell data. So now that we want to go into spatial omics, from single cell to spatial omics. So I like this picture that I saw from Twitter a few years ago that interestingly summarize the features of different type of technology. So bulk is really a heterogeneous mixture of different types of cells in the system. So although using single cell technologies, we can separate or cluster different cell types or different cell states of cells into different clusters, but we do not know their activity or relative spatial distribution in the actual tissues in the real life physiological conditions. But the spatial omics can indeed give us this information, not only characterizing different cell types, but also given their spatial information so that we can use this information to reconstruct the original tissue as well as study these physiological conditions in different tissues and disease samples. So yeah, so we recently in collaborating with Dr. Jie Sun, also our colleague at UVA, to use spatial transcriptomics data to study the long tissue samples after the long COVID infection. So in this recent work that spearheaded by my now a research assistant professor, Sean Hu, in my lab, in collaborating with Jason's lab, and in this work recently published in Nature, we identified a very interesting pattern that could potentially explain the molecular mechanisms of why some people will grow a very long COVID compared to the short or acute infection of COVID disease. So in this spatial transcriptomic data, we use different colors to represent different cells. The idea is that in this human lung infection, after a lung infection, there could be the existence of some immune cells, namely CD8 plus T cells, that are still existing in this tissue, and with the KRT5 plus and the KRT8 plus, which are the progenitor cells that could potentially prevent these normal epithelial cells from regrowing. So in this case, these in terms of mechanism, these CD8 plus T cell, they could derive from the interferon gamma and the TNF simulated lung macrophages, and they can chronically release IL-1 beta that maintains these KLT8 high transitional and ectopic KLT5 basal cell progenitors, and this could just impairing the regeneration of the tissue. So after this infection, so this is just an example of show the power of directly using spatial transcriptomic data with identified gene signatures to identify the potential cell types to explain the mechanism. So with these single cell or spatial omics data, we also wanted to see how we could predict active transcription factors from such spatial omics maps. So this is still an unpublished ongoing work spearheaded by my first year graduate student, Jingyi Wang. So now we have the BART-2 available here, but the idea is to see how we could generate the correct input data for BART to be able to work to predict the active transcription factors. So most commonly used spatial tools are spatial transcriptomic data, in which we have the gene expression profiles in the usually a two-dimensional spatial map. So we wanted to identify the goal is that we wanted to identify the transcription factors that could be active in driving the spatial variation of different cell types or cell differentiation, or just cell gene expression pattern, gene expression differences across a spatial pattern. So in this way, what Jingyi tried is that we wanted to identify a gene set that have both features. So the first feature is that these gene sets is co-expression pattern should follow some type of a continuous trajectory from these spatial data. And the second is that the expression of this gene set should be spatially variable across this 2D spatial map. So then we can identify these differentially expressed genes along this trajectory, or we call the trajectory associated genes, and also spatially variable genes. And we either get this, we can get either overlap or the union of these these genes as an input for BART. So there are several groups that also developed spatial attack technology. So similarly, we can also use the same idea to identify spatially variable attack regions or differential attack regions and attack regions that are differentially follow as a certain trajectory used as input of BART to predict active transcription factors. So then we wanted to study use a few case studies to show the performance or the power of BART. Here we call this about BART-spatial, to see how we can use this to study gene regulation from spatial data. So we use one data set recently published also in mouse brain spatial. The unique part for this data set is that they have both spatial transcriptomic data as well as spatial attack data from the same system, so that we can take advantage of this data to make some validations. So we specifically focus on two cell types, the differentiation from the radioglia into the post-mitotic premature neurons. And we use the annotation directly from their publication, their published annotation, that we can see this dark purple cell for radioglia will be differentiated into post-mitotic premature neurons in this show this spatial pattern. And by either using their RNA information or using their attack information, we can generate a trajectory to show the continuous gene expression pattern changes. Here it shows as in the pseudotime. And also we can see some genes with transcription factors, they will also show spatial variability and spatial differences between these two cell types. And now we're going to track whether BART can help us predict the transcription factors active in this during this cell differentiation process. So so first, because most of available data and widely applied technologies are spatial transcriptomics only. So we use the spatial transcriptomics part alone to make the prediction. So as I mentioned, we identified over 300 differentially spaced genes that are differential and associated with the trajectory, and also more than 500 differential genes that are show the spatially variable patterns. And using the 146 genes that are in the intersection as the input for BART, we can identify the list of transcription factors as the BART output. So we are happy to see that the highest ranked SOX9 is a known transcription factor in this system, as well as several other transcription factors, including neural D2. That these transcription factors, they're also known factors in this specific system. And just as an example, including the known SOX9, as well as several other predicted one, SMART-B1 and the KMT2D, these we show that the expression level of this transcription factor coding genes itself also show some spatial variable pattern. But we're not only interested in identifying those transcription factors that also show the spatial variable pattern itself. We wanted to validate whether the BART predicted transcription factors that rank on top of the list, whether they are real or not. Other than SOX9, which is the top first known factor, then we look at the second one, KOI4, ranked number 2 in this prediction list. So interestingly, although we found that KOI4 itself does not show a spatial variable pattern in terms of their own expression, but we found that the chromatin accessibility regions, taking advantage of the spatial attack information, the attack signals that have KOI4 motif indeed show a spatially variable pattern. And we see that this KOI4 motif is enriched in this spatially variable accessible chromatin regions that identified from the spatial ATAC-seq data alone. And also the spatial variable differential ATAC-seq regions that have a KOF motif, that have a lot more KOF motif compared to other non-spatially variable differential ATAC-seq region. So this example indeed show that the transcription factors activity itself is not always correlated with the expression level of the transcription factor coding gene. And especially in this spatial omics data, the KL4, even if they do not show a spatial variability across these different regions, their binding sites, their potentially binding sites, here defined as the chromatin accessible regions that contain this motif, will also show a spatially variable activity pattern that regulates their potential target genes. So although the available data are not that many, we also show that using BART-spatial tool, we can also predict transcription factors from the spatial ATAC-seq data. And using similar ideas that I mentioned to identify the union regions in this case that are differential across the trajectory, as well as show a spatial variability, and we can predict the active transcription factors and using the target genes showing their relative enrichment for the functional terms, which indicates that these predicted transcription factors are real. And also in this particular case, we show that using gene set or spatial transcriptomic information or a region set, which are spatial ATAC information, their prediction results of active transcription factors are consistent, which also indicates that our prediction is reliable and robust. And these labeled red one are those ranked on top or identified as significant from both transcriptomic data as well as spatial attack data. So in summary, I hope I showed you that such emerging single-cell and spatial omics technologies can accelerate our biomedical research discoveries. And using computational modeling, especially integrating information from the public domain, can help us transform the high-throughput data into biological knowledge. And I show you the series of tools we developed, the BART series, and specifically the two versions of BARTsc for predicting active transcription factors from single-cell omics data. And the BART-spatial is an ongoing work that we are developing to predict active transcription factors from spatial omics data, spatial transcriptomics, and or spatial ATAC-seq data. And with that, I would like to thank my lab. So without them, we cannot do any of those work. And I already mentioned that the work that I presented today are done by Shuang Hu, Hongpan Zhang, Jingyi Wang, Zhenjia Wang, and Wenjing Ma, as well as the helpful input from all the current and previous lab members. And I also want to thank my collaborators here at UVA and across the country who not only provide us with their valuable different type of data for us to dig into, but also their helpful discussions about biological scientific insights is the ultimate motivation for us to develop new computational tools to advance the biological discovery. And of course, I want to thank the funding resources that support my lab's research. So, all right. With that, I would like to end here and answer any questions you have.
Chongzhi, that's fantastic. What a huge amount of work. It's really, really beautiful stuff. Thank you so much for sharing that with us here at our 150th seminar series installment. So thank you. I want to ask you something about generalizability, reproducibility, and sample size. Now, obviously, with any of these single cells, we can like take every individual cell, as many as we want out of the animal, I suppose, and we can make measurements on them. But I think one of the things you showed is some of the work that you were doing with some of the pancreatic cases where you had like 4 of one type of a mouse and 4 of another type of mouse. What sort of considerations I know that these are genetically modified mice and such, but what sort of consideration does one need to take into account in terms of the overall sample size?
That's a great question. So just as any biomedical or biological studies, the sample size and generalizability need to be taken advantage of. So, but I want to bring up the point that because the idea of BART series, we only use the information we integrated from the public domain. So the sample size and the generalizability will mainly rely on the input data, namely which in the specific cell systems or biological systems that you are studying. So then by the pre-processing or analyzing those the data, what BART really needs is just a gene set or a region set. It's the simplest form of type of input without the sample or cell type specificity or system specificity. So so BART is able to just use the common information from the gene sets or region sets to make the connection and to make the reasonable prediction. Yeah.
Are those reproducible? If you like said had say 4 animals and you gave your BART the information it needs, and then if you took 4 more animals, would you get the same result?
Yeah, good question. So in that specific examples, we actually have for the mouse PDX studies, we have 2 individuals, And of course, there are commonly shared results as well as specific results. So I think an important question is if these data are from different individual, and whether the question is whether we want to identify the common pattern that could show that there may be some common mechanism in this disease, or the sample specific information could either be contributed by just the sample variability or the individuals, because we know different patient, different individual are so different. So in that case, we do need to maybe have more samples from the same individual or to evaluate that.
Is it a cost related thing that kind of puts a governor on the number of animals that you can draw from, or is it just like a practicality?
I think it's mainly the cost thing. Yeah.
It
Ideal. Yeah.
always boils down to money, right?
Yeah, indeed, indeed. As well as a lot of the single cell studies. Although some people may think that in the single cell omics, single cell study, or even spatial omics, a spatial spot can be one sample, or a cell is one sample. So the sample size is no longer an issue. But it's yes and no. Because in this specific type of data, we want to develop, the conventional sample size analysis models and approaches may not work anymore because single cell carries their own features, and one may need to develop more tools to better evaluate the sample size in these studies.
Well, that's great.
Spatial.
Yeah, because it leads into my final question. In our last few minutes here, I wanted to say, or have you kind of state, like, what data science methods and approaches do you want to see more of in multi-omics research? What more would you like to see? What can data science do for you?
Absolutely, that's a great question. So I think my biased view or my preference is I always wanted to see data science models or AI models that are that really can help us dive into specific biological questions, or models that are customized designed to really address a useful or meaningful biological question, and also an interpretable or explainable models. Because the ultimate goal of all of our biomedical research is to make sense from the data. Is we want to explain why. We want to explain the underlying rules and mechanisms of all the patterns we observe. So model interpretability and model explainability is extremely important.
That's wonderful, Chongzhi. Thank you so much. We really look forward to you being a mentor at this year's Biomedical Data Science Innovation Lab, where I'm sure that many of our participants are going to have lots of things to talk to you about, and about your tools, about where there might be gaps, where they could help fill them, and certain interesting applications for this type of work in clinical trials, and to help drive hypotheses that we may be able to further test. I want to thank you for being the 150th installment in our Foundations of Biomedical Data Science seminar series. I want to give one more thank you to Gina Kamdar and to Hannah Kears for all of their hard work to make this possible. I want to thank the National Institutes of General Medical Sciences for funding our efforts. We look forward to being able to bring this to you for a long time into the future. Have a wonderful Friday, everybody, and we will see you next time. Thanks again, Chongzhi.
Thank you so much. Thank you so much, Jack, and thank you everyone for your attention. And yeah, and thank you, Gina.